Welcome to another video. This time we'll be focusing on antibody structure and function. Let's talk a little bit about some student learning targets for today. By the end of this video, you should be able to understand the structure and function of an antibody, explain the process of making antibodies, and correlate concepts of intermolecular forces and some thermodynamic principles to the bonding of antibodies to antigens. Let's move on. Let's talk a little bit about what antibodies are. Antibodies are simply blood proteins that are produced by specific cells in the blood called B cells, and they respond and counteract specific antigens. Antigens are things that we typically don't want inside of the blood. Typically, these are things like viruses and bacteria. This is a major component of the human immune system. One thing to really keep in mind, though, is that antibodies are specific to antigens. What that means is that an antibody that works for one specific antigen will not work for another one. And we'll talk about why that's the case here in just a couple of moments. Let's talk a little bit about the structure of an antibody. An antibody is a Y-shaped protein, and most of the Y-shaped protein is actually similar across nearly all antibodies. The key part that's different, as you see in the image, is the hypervariable region, which is located at the top of the Y shape. This region differs based on the antigen to which it attaches. So this is where the specificity of the antigen is very important. In this variable region, that can change based on what type of antigen we're looking at trying to attach to. So you could argue that it's antigen specific. Now, antibodies function in a variety of different ways once they've attached to the antigen. One way is neutralization. The antibody attaches to a part of the antigen that ends up halting its infectious ability. Typically, these are spike proteins that may be found on the outside of bacteria and viruses. Agglutination and precipitation is where antibodies help bond antigens together into very large molecules that are then destroyed by white blood cells through the process of phagocytosis. All processes, though, require the bonding of the antibody to the antigen, so that bond that is formed between them is very, very important. If an antibody cannot be produced to be able to bond to the antigen, that's very bad news for the organism. So let's take a look at an image here. Just a couple of ways. If we look on the left side of the image here, this is a typical virus that attaches to proteins on the outside of the cell. The cell doesn't recognize it as anything infectious, so it allows it to go inside of the cell. The virus then releases the RNA to be used in the process of protein synthesis inside of the cell, thus resulting in more viruses being produced. However, on the right-hand side here, we have a couple different ways that the antibodies actually work. The first one, kind of on the top left there, is blocked attachment. So the antibodies bond on the outside of the virus, but either the cell does not recognize that and doesn't allow it to go in, or endocytosis is actually blocked. It can also block the ability for the RNA to be released inside of the cell. Notice at the very top right, the antibodies are bonding to multiple viral structures, which then could trigger a white blood cell to perform phagocytosis and destroy the viruses. So antibodies actually work in a variety of different ways to prevent infection from occurring. So what's important here is the effectiveness of the bonding. The effectiveness of the antibody is directly correlated to how well it bonds with the antigen. A suitable antibody will fit like a lock and key with the antigen. This is very similar to how an enzyme and substrate work. Hopefully you remember that from biology. The better fit and stronger intermolecular forces that occur within the antigen-antibody complex, the more likely the antibody will initiate an immune response. So that connection, that bond, those intermolecular forces that are occurring between the antigen and the antibody are crucial in the antibody actually helping to signal for an immune response inside of the body. The epitope is the antigen part that bonds with the paratope, the antibody part. The locations of individual atoms and functional groups on both parts determine what types of bonds are being formed. Most bonds are either ionic, hydrogen bonding, or London dispersion forces, which we've talked about quite a bit in class. So if you look on the right-hand side here, the individual parts, the individual atoms, and how well they fit together and how strong those forces are that are found in between the antigen and the antibody determine whether the antibody is going to actually work or not. Now remember, we are forming bonds here, and forming bonds is an exothermic process. So you can see a variety of different interactions that are occurring between the antigen and the antibody. The stronger those interactions are, the more likely the antibody will attach to the antigen and therefore result in an antibody that fully functions. So how are antibodies created? Well, this is actually a fairly complex process, and without getting into too much detail, antibodies are made by B cells. B cells can only produce one type of antibody. So if I have a single B cell, it is only going to be able to produce one type of antibody in its lifetime. Different B cells can produce different antibodies, but a specific B cell can only produce one type over its lifetime. To start the process, you have what are called naive B cells, and these are presented with the antigen by the T cells. This initiates the production of antibodies through the process of protein synthesis inside of the cell. Again, it's very, very similar to what you would see inside of a normal cell. The only difference is that they will make that specific protein, that specific antibody, in that specific B cell. What's nice is that once you have this infection, once you have produced the antibody, the B cell will remain inside of your body. And so the B cell will continue to either A, produce those antibodies, or B, have the potential to produce those antibodies. That's one of the reasons why they always say if you get infected with a certain virus, you're likely not to have it again, because you will either be able to have the antibodies already inside you, or the B cells will be able to create those antibodies much faster than what they would if it were a novel or new illness found inside of the body. So lots of really interesting and unique applications to a lot of this material. So hopefully you understand just a little bit about how antibodies function inside of the body and the process of making those antibodies, and then be able to correlate some of those concepts and the importance of intermolecular forces and thermodynamic principles to the bonding of antibodies to antigens. We're going to expand on this a little bit more by looking at how antibodies can be used in technological applications outside of just curing infectious diseases. Thanks for listening. Have a great day, guys, and we will talk to you later. Bye-bye.